My name is Aymar. I'm a PhD candidate at the São Paulo State University in Brazil. And today I'm talk about some results from my thesis and a work entitled Impacts of Invasive Fish Species in Neotropical Reservoir. So let's go. So first of all, I will bring some concepts and implications of some terms. And here I'm talking about the cause of invasions. In this science, it's about the population dynamics and impacts of species in a geographic space outside their natural distribution range. Of course, by anthropic actions, being intentional or not. And this subarea of the ecology of invasions, it's growing fastly and largely all world because we see more of the impacts of the introduced species on the biodiversity and economically terms. So obviously we'll study more about this. So what is native species? Within its natural distribution range. For example, when we talk about the javali, Suliscrofa, this species it's native of some parts of Asia and Europe. But when we talk about the same species in Brazil, for example, we can talk about a non-native species because it is outside its natural distribution area. So when we talk about a non-native species, we talk about a potential invasive species because if this species establish in this new environment, treating or altering the local biodiversity, we can talk about invasive species, for example, the same javali in some areas in Brazil, because the invasion is a process that comprises from the transport to the impacts. For example, the transport, the introduction, the establish of the population, if the population spread can bring some impacts, and these impacts, when we have the perception, almost then it's the high impacts because this high impacts more it's more measurable. And when we talk about invasion in Brazil, we talk about a large country, a large areas. For example, we have many large rivers, and these rivers have a large number of dams, creating new environments, new reservoirs, creating new environment, new limnological conditions, with, of course, the community response to this. And all these dams are spread in all Brazilian biomes, and particularly the Atlantic rainforest. We have the Paraná River, and this hydrographic area, the hydrological regime is regulated by the 26 large reservoirs. It's one of the most regulated river streets in the world. And of course, we have an area with large human use. We open doors for many vectors of introducing. For example, the Upper Paraná River, we have a predator fish species that are introduced since the 70s. For example, the silver croaker, peacock bass, and dorado. And recently, we have the biggest fish of South America, the pirarucu, which is native from Amazon basin. We have most of the studies concentrated on predators, but and fish species which occupy the low trophic level on the food chain. We have few studies which have elucidated about the possible effects of small or medium non-native fish species. For example, we have the 𝙁𝙗𝙤𝙪 veni, which have high evidence and dominance in the Upper Paraná reservoirs. This species is native from the lower and middle Tocantins River and some Amazon River tributaries. And we have few evidence about the vector of the introduction and the year of introduction, but the most probably vector was the aquarium trade. And previously, this species was reported as 𝙁𝙗𝙤𝙪 proximus in Paraná River, but after some genetic studies, they confirm that this species is 𝙁𝙗𝙤𝙪 veni. And the presence of this species has been suggested since 2007. And this species is also reported in Argentina in 2015 by the Paraná River Basin, connecting the rivers intercountry. So here we investigated the changes in the diversity of fish assemblage as a possible result of the invasion of the 𝙁𝙗𝙤𝙪 veni. We hypothesized that the invasion altered the composition and the structure of diversity by taxonomic, functional, and phylogenetic indices on the fish community in these reservoirs on Upper Paraná River. And for this, we use datasets of long-term of monitoring of fish fauna over 15 years of study in Upper Paraná River. And these reserves are larger and connected each other by artificial channels. And this fish fauna monitoring consists in 3 monthly sampling in 3 or 4 sample sites for each reservoir. So for each sampling site, we calculated the indices of the fish fauna structure, such as the taxonomic, functional, and phylogenetic indices per year. For taxonomic indices, we use species richness, for the functional richness, and phylogenetic richness. For the phylogenetic richness, we take the molecular sequence from the gene bank for each species. And later we done generalized linear mixed models using the indices has response variable, and for explanatory we use the invasor abundance, and using for random effects the year and the reservoir. Also, we test with the fish fauna composition change by the time size impoundment and by the 𝙂𝙬𝙤𝙪 vein abundance, or by the reservoir, which is the water parameters. And also we did correlation with the potassium competitors identified by phylogenetic and functional traits by cluster. And we did negative binomial with zero inflate models using the abundance of the competitor and the abundance of the 𝙂𝙬𝙤𝙪 vein. Bringing the results, we did here the population dynamic of 𝙂𝙬𝙤𝙪 vein by the years. Chronologically, we have the first records in Ilha Solteira Reservoir, and after the Tietê and Jupiá Reservoir in 2001, and Porto Primavera Reservoir in the next year. And in all reservoirs, we saw a boom of the increase of the abundance, and later we saw a population more stable. However, we don't found negative correlations about the 𝙂𝙬𝙤𝙪 vein abundance and the indices of the fish fauna structure, and the same pattern we found by each reservoir. But the abundance of 𝙂𝙬𝙤𝙪 vein contributes to changes in the fish fauna composition. Also, we saw that the time since the impoundment and the water conditions also contribute to change in the fish fauna composition. And by the phylogenetic tree and the cluster of the functional traits, we found two potential competitors of 𝙂𝙬𝙤𝙪 vein. They are 𝙂𝙬𝙤 brasiliensis, contender native from Paraná River Basin, and Satanoperca papaterra, a non-native fish species, but very close phylogenetically with these other two species. And by the binomial negative models, we found that the 𝙂𝙬𝙤𝙪 vein have a negative effect in the Satanoperca papaterra when the 𝙂𝙬𝙤𝙪 vein abundance is high. And for the native species, we found two negative effects of 𝙂𝙬𝙤𝙪 vein decreasing the abundance. So comparing the temporal population dynamics, we can see that the native one in black is probably more negative affected or different affected by 𝙂𝙬𝙤𝙪 vein than the Satanoperca papaterra in red. And the results which I showed probably is driven by the generalist behavior of 𝙂𝙬𝙤𝙪 vein. 𝙂𝙬𝙤𝙪 vein is a bottom feeder. The diet consists mostly invertebrates like mollusks, gastropods, or larvae of insects. Also, the 𝙂𝙬𝙤𝙪 vein have a parental care. He's a territorialist species, and the capacity to occupy and overlapping many niches favor this species to be successful in the process of invasion. And species phylogenetic and functional nearest related can lead to resource partitioning and overlapping in life history characteristics. And due to this, the invader can affect the distribution of the native competitor, decreasing their evidence or displacing from the ideal habitats through the competitive exclusion. And it's obvious that the consequence of invasions across the biodiversity sphere and can lead to economic and social loss. So it's clear that the effects of the fish invasion on the structure and composition of assemblage in freshwater system still required further analysis, because almost the studies are of the short time duration, like 1 or 2 years, or they don't have the records of the communities since the species was introduced. And to finish, of course, I would like thanks for the organization to give me this opportunity to talk about my work, to talk about some results from my thesis, my university in Brazil, my lab here in abroad in Valencia, which nowadays I'm doing an internship. Yeah, of course, would like the FAPESP would give me the grants. So I gave my email in the first slide, and be free to write to me if you have any doubt or any curiosity about my work. Okay, so thank you everybody. and enjoy the event. Bye bye.